Good evening to everyone. Myself Revati Dharampal Sagare, PhD Research Scholar, KLA College of Pharmacy, Hubli. Today I am going to present a poster presentation on the topic Design, Optimization and Characterization of gamma-MPS doped halloysite nanotubes by Ultraturrax Homogenizer. Halloysite nanotubes are naturally occurring one-dimensionally aluminum silicate minerals with an empirical formula Al2Si2O5OH4 2H2O. These are natural, biocompatible and biodegradable. Since they are having a unique and versatile properties, they have a wide range of applications in both biomedical as well as the non-biomedical science such as gene delivery, cancer isolation, tissue engineering, cosmetics and wastewater treatment. An alternative to these advantages, HNDs have some shortfalls like insufficient mechanical strength, lack of drug loading capacity, and lack of drug polymer dispersion ability. To overcome these drawbacks, the interface between the nanotube and the polymer must be engineered by the doping process. Objective is to design, optimize, and characterize halloysite doped polymer conjugate by homogenization technique using gamma-MPS as a silane coupling agent. Materials and methods. Since the independent variables were continuous with numerical, hence custom design was chosen to investigate the effect of independent variables at two different levels on the response variables. Results and discussion. Doping of pristine HND was successfully confirmed by the presence of functional group characterized by the FTIR analysis. The spectrum of pristine HND revealed the two absorption peaks at 910 and 1031, which signifies the presence of eliminol and the selenol groups. Whereas the two new peaks were observed into the doped HNDs, one at 1497 and the other peak at 2967, which confirms the presence of the deformation of the CH2 groups and the symmetric stitching of the CH2 group. The XRD analysis of pristine HND revealed a sharp narrow peak at 11.36 degrees with the basal reflection of 7.47 Armstrong, signifying that it belongs to the state of hydration. Upon doping, the F7 sample exhibited a slight broad peak to a higher 2 theta value of 12.07 degrees, and this is achievable by the influence of speed and the temperature generated during the homogenization technique. Whereas the F8 sample, we could observe it exhibited a sharp narrow peak but at the lower 2 theta value as compared to F7 sample at 11.68 degrees, and this indicates the slight doping of pristine HNDs. Halloysite nanotubes are identically characterized by hollow nanotubular structure with an open-ended lumen, with the lengths ranging from 400 to 1000 nanometers. Upon doping the F3 samples, we could observe that the length of the nanotubes was enlarged to 600 nanometer and a layer of gamma-MPS was wrapped onto the surface of HNDs as the surface had become the rough. The main observation that we could find is the F5 sample, wherein the aggregation of the nanotubes is due to the presence of the van der Waals forces. Zeta potential analysis, the pristine HND revealed a negative zeta potential value of minus 33.64, and this negative charge is mainly due to the presence of the silica groups on the halloysite nanotubes. All the doped HND exhibited the higher zeta potential values. The effect of independent variables on the response variables exhibited a synergistic effect as depicted in these 3D surface plots. The results of ANOVA studies confirmed that the effect of independent variables exhibited a statistically significant as the p-value was less than 0.05. These optimized dope HNDs were characterized for the SEM analysis, wherein we could observe the pristine HND at 100x, the nanotubes were small with an uneven morphology appearing as a cotton cloud of the nanotubes. But upon doping at the same magnification, the intensity of the aggregation was drastically reduced and the nanotubes were clearly observable within proper dimensions. Thus, we can conclude that halloysite nanotubes were successfully doped with gamma-MPS using Ultraturrax homogenizer, and the results of statistical analysis indicate that the application of custom design proves to be a statistical and ideal tool for optimizing the doping of halloysite nanotubes. The research work was supported by the funding agency AICT RPS scheme under the Government of India. Thank you.